Okay, everybody. Welcome back. Don't expect anyone today, but if I do get someone, that'd be great. Today, we are going to be working on chapters 14A and 14B, so please get those ready. We'll be starting here in a moment. Okay, we're talking about sensory physiology here, how the body senses discrete sensations. Okay, what are some general properties of sensory systems? Well, we talk about the stimulus source. Where does the stimulus come from? Well, it's internal, it's within the body. That means something's happening inside of your body and you're sensing it. External would then be something happening to you from the outside, so surface stimuli generally. Another type of way to distinguish different types of stimulus is the energy source or type. So for example, force would be touch, temperature, hot and cold, light, vision, pressure waves, that's hearing, believe it or not. We are going to be talking about how pressure waves are sensed by the ears, and chemicals, which would be smell and taste. Where do you find receptors? Where receptors are found in sense organs, which receive the stimulus and act as transducers. The idea is the stimulus is not going to be in the form of action potentials. So the sensory structure, sensory organs, transduce whatever stimulus it is, as we saw above, into action potentials, a series of action potentials. Now, if you remember, we talked about how intensity is coded for. So please try to remember what we talked about with that. I'll be repeating it again today, but make a note of that because I'm going to be asking you to tell me about that on an essay. Okay, afferent pathway. What does afferent mean? It means towards the central nervous system. So this would be sensory. Afferent pathway signals move through sensory nerves towards the CNS, which would be either the brain or spinal cord. Then at CNS integration, be it the brain or spinal cord. By the way, if it's spinal cord, it'd be for reflexes, and any other kind of integration site would be for senses other than reflexes. So there you go. All right, what are the different sensory receptor types? It can be categorized by structure. First type is simple. And what that means is the simple receptors are more or less a neuron. We talked about how neurons are built, the cell body, dendrites, and axons. So really simple receptors are just regular neurons. And the two types of senses that use free nerve endings are pain and heat. Another type sensory receptor would be complex neural, where the nerve endings are enclosed in connective tissue capsules. So for example, pressure is sensed by the Pacinian corpuscles, and the tips of those receptors are wrapped in connective tissue to reduce their sensitivity, believe it or not, so that you only feel deep pressure and ongoing sensation. The special senses are very unusual because although they vary in the type of structure, the type of cell that's used, the special senses are special for a couple of reasons, and I'll explain it when we get to that. That'll be in our next lecture coming up in just a moment. So the idea here is the receptors pass the signal to afferent nerve and do not have axons. So that's for most of these. Now, this is really weird because these special sensory receptors are not regular neurons. They don't even have action potentials. They have what are called receptor potentials. And we'll be talking about that. So your special senses, vision, taste, smell. Smell is a little bit different than the others, but hearing, hearing, taste, balance, etc. Those are different than our other senses because they don't have regular neurons as their receptors. Even modified neurons, they have special sensory receptors that convert the stimulus into receptor potentials and then immediately release acetylcholine or stop releasing it in the case of vision. I'll be telling you about that when we get there. So another category can be by the function or stimulus type. So one type of receptor would be chemoreceptors, and that is they sense chemicals. And an example of that is an oxygen and pH sensor in the brain. Another example would be smell and taste. Those are all receptors that pick up chemicals, either in your blood or in your saliva or in the air. Now, mechanoreceptors detect physical changes in the tissue. So that means the tissue has to change shape or pressure has to be formed on the tissue. So for example, touch, muscle length changes, joint angle changes are mechanoreceptors because that's a change in the physical shape, angle of the structure. Thermoreceptors are really weird. Well, there's a lot of weird things in sensation, but what's weird about them is they detect changes in the temperature normally of your skin, not constant temperature. So both heat and cold receptors don't test or sense overall temperature. They sense whether your skin is getting warmer, colder. That's why people who have cool hands feel like their hands are normal. People who have warm hands feel like their hands are normal because they're not changing temperature. I'll be talking a little bit about the cannibal pot phenomenon and why we sense certain things in bathtubs, believe it or not. Okay, the last type is photoreceptors. Photo means light. It doesn't mean photograph. Photograph literally means an image made from light. So photo just means light. So they detect light in the eyes. And we're going to be talking about why is vision so much different than the other senses. It's backwards in two separate directions. And the two types of photoreceptors that you find in the eyes are rods and cones. We'll be talking about similarities and differences between those photoreceptors coming up. Okay, this is an image directly from your text, shows a sensory cell. This is one that has a pseudo-unipolar, but don't need to worry about this. The key is that we have free nerve endings, dendrites. They're going to be bringing the sensory signals in, going through the axon, bypassing the cell body, and eventually making their way out to the end. Now, notice that in this case, these were free nerve endings, like heat, pain, but in pressure, for example, the Pacinian corpuscles, you have an encapsulated nerve ending, which makes it more difficult to stimulate and gives constant pressure. And the last type is the specialized receptor cell. These are special sensory cells. Notice this is a rod. We're going to see the similarity. Cones are pointed at the end, but notice that there is no axon. It's just the cell body, and then a little process that sticks out, no axon, no action potentials, very strange. All right, special senses. They almost always pick up external stimuli. There's one that's the exception, but almost always it's something coming into you from the outside. Even taste is an external stimulus because that food came in from the outside into your mouth. So vision would be your eyes, hearing, ears, taste, tongue. We're actually going to be answering the question later on, if a tree falls in the forest, there's no one there to hear it, does it still make a sound? I'll answer that for you convincingly, hopefully. The taste is the tongue. We're going to be talking about the difference between taste buds and the papillae on the tongue. They're not the same thing. So those little bumps on your tongue are not taste buds. Anyway, smell is the olfactory epithelium, not your nose. So the idea here is this object sticks out of the front of your face that we call our nose, has virtually nothing to do with your sense of smell. The olfactory epithelium does, and that's found up in the roof of your nasal cavity, not in your nose. Okay, internal stimuli, equilibrium. And this is really different because it gives us our sense of balance. So spinning balance as well as our sense of gravity. So spinning balance and gravity sensors are both internal stimuli. Okay, now let's take a look at this figure, and I'll talk to you all about it. I may be asking you about this both as a regular question and or extra credit, so take lots of notes here. Okay, now we're going to look at the cerebral cortex. The cerebrum is inside your skull, and you can see the wrinkly part. Most people are familiar with it. Well, if we take a section of it and measure what happens when you stimulate that part of it, let's say we just take a section right through here. If we take a section right through here and we stimulate with an electric probe, what happens is, if we're doing this with an open cranium, the patient feels like they're being stimulated. Let's say, for example, I stimulated this part of the brain. You would believe that your midsection had been touched. You have no way of knowing whether or not it was actually touched, other than the fact that this part of the brain is being stimulated. Now, normally, these direct pathways come from your midsection right out to this spot on your brain that codes for your midsection. Interestingly, the way that it's set up doesn't make perfect anatomical sense. Your face is separated from your head. Your genitals are separated from all the rest of you, interestingly. So, for example, genitals are here, down deep in this groove, then feet. Now, notice how large the hands are. What does this mean? It means that a large amount of your cerebral cortex, lots of your brain, and lots of your sensory cells in your skin are dedicated to your hands. That's why the hands seem so big, because they're covering so much of this. Here's your face. Notice how big the lips are. The lips are the most sensitive part of the face. And then we have our jaw, believe it or not, that's separated, and then the tongue down below that. What does the thalamus do? It relays signals coming up from the spinal cord out to the part of the cerebral cortex. Now, if we take this entire map of how much of the body is dedicated, how much of the cerebral cortex is dedicated to different parts of the body, put it together, we get what is called the homunculus. And this is the homunculus man. Now, what does the homunculus man mean? So if I ask you, what is this guy showing? Well, he looks pretty silly, doesn't he? But what it means is the size of the various body parts correspond to how much of your cerebral cortex is dedicated to that body part, and therefore how many sensory cells there are in the skin or other surface structure dedicated to it. So you can see ears are sensitive, lips are very sensitive, tongue is sensitive, and the hands are exceptionally sensitive. This is why we read Braille with our fingertips and not with our back or our feet. Our feet are just not that sensitive. But the question is, how sensitive are our genitals? Well, we believe them to be very sensitive because stimulus there elicits a strong response. But believe it or not, there's not that many sensory receptors in the genitals as compared to the lips, tongue, and hands. So the question I asked is kind of a little bit tongue-in-cheek, no pun intended, a little bit joking, is if you didn't have any hands, could you use your genitals to read Braille? I know that sounds disgusting, but could you? And the answer is no, there's just not enough sensitivity there. But what body part could you use? Even though it's not very sanitary, you could use your lips or tongue to read Braille in the absence of hands, because look at how sensitive the lips and tongue are. And that's one of the reasons scientists believe humans really like kissing, because you're touching each other's lips, which are both very sensitive, and each other's tongue. So there you go. If I ask you about the homunculus man, talk about what I just told you, take detailed notes. And if you didn't take detailed notes, rewind me and retake those notes. I may be asking about this. All right, sensory modality. So location. Now, remember I said it's where you believe a sensation has come from, has to do with where in the brain it's stimulated. So here's what's interesting. There's no information in the action potentials themselves that says where that information came from. It's the labeled line phenomenon. That is, there's a direct connection between a part of the brain and a body part that's sending signals. And we learn over time that that part of the brain is dedicated to that part of the body because when it's touched, we feel it in that part of the brain. And that happens way before we're born. All of that mapping happens. So if I asked, is there any information in the action potentials themselves that says where a signal came from? The answer is false. It's the direct physical connection between the skin and all the way up through the vertebral canal, spinal cord, thalamus, and eventually the cerebral cortex. So it's that direct connection that tells you where it's from, not how it's coded for. And that's very different than cell phones. Cell phones are not physically connected. So what tells the cell phone tower which is your cell phone and which is somebody else's? That in that case, it is the pattern of signals coming from your phone that tells Verizon or whatever you're using that it's your phone and not somebody else. But we're old school. Our bodies are old school. It's direct connection, just the way the old landlines. The phone company knew where you were by the physical wire connection between your phone, your house, and the phone company. All right, location determination exceptions, sound. Now we're going to be talking about how do we determine sound. Obviously, it can't be from what body part is being stimulated because the inside of our brains are being stimulated. Well, there's two ways that we determine sound source. And I'll be talking about that when we get to how hearing works. All right, now this is the restatement of what I talked about before when we talked about how do we determine intensity. How intense is the stimulus? Well, it's determined by how fast the action potentials are coming. If you remember, action potentials do not change in intensity. They're always 100 millivolts. So if you can't use how intense the action potential is, How do we determine how intense the stimulus is? It's how many are coming. It's very much like Morse code. So for example, if a very light sensation is picked up by our skin, what you would see in the nerve is blips like this. Blip, blip, blip. Now this is slowed down. But what if it was a really intense, so you're pushing very hard on that part of the skin? It would be more like blip, 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 blip. And that would be the code that your brain would use to determine whether it was a very intense pressure or a less intense pressure. It's the frequency, how many per second come, not the intensity of the individual action potential. So try to get that into your cerebral cortex. All right. Now, in terms of duration, we're going to be talking about what happens if the stimulus lasts for more than a moment. Other than pain, the perception of stimulus is diminished over time if there's no changes. Why is this the case? Well, your brain really wants to know, besides pain, is the stimulus changing? Is there something new happening? So rather than bombarding the brain with a constant flow of information, the brain really wants to know, is the smell getting any greater, any lesser? Is there a new smell? Is the pressure that's on our hands getting greater or lesser? Now, we're going to be talking about olfactory fatigue in a minute here, and why you should never stay in a room when you smell natural gas. Now, olfactory fatigue, I'll talk about it right now just in case it doesn't come up. So what is olfactory fatigue? Well, olfaction is smell. I think it is going to come up again, but if it does, I'll repeat myself, and that's fine. Smell. Fatigue means getting tired. So what is olfactory fatigue? Well, you may have noticed that when you first walk into a room that you're unfamiliar with, it's not your house, you smell things more intensely than if you stay there. Well, why is that the case? Well, your brain really wants to know that this is a new smell. And so it sends lots of information to your brain. Hey, I'm smelling some new things. And if you stay there for a while, the odor molecules don't get less, but you send fewer signals. Now, how does this affect our safety? Well, if you walk into a room and smell natural gas, which, by the way, both natural gas and propane have been enhanced with a smell molecule called methyl mercaptan. Now, what is interesting about methyl mercaptan? Well, it burns with the natural gas and propane, but it has an odor that says, hey, it's dangerous to be in here, get out. Now, the problem is if you stay in a room when you smell natural gas, olfactory fatigue kicks in and you stop smelling it. What could happen? Well, the gas could overpower you, but more likely, you would walk across a carpet, a little bit of static electricity is all it takes, a tiny little spark, and kaboom. And that actually happened to some restaurant goers a few years back. They smelled natural gas. They told the waiter, they told the manager, they didn't do anything. Everybody went through olfactory fatigue. Adaptation kicked in. They stopped smelling it. Small spark happened. The whole place exploded. So olfactory fatigue can kill you if you don't take seriously certain odors. That's a good example. All right. Okay, so we're going to be talking about the receptive field phenomenon. We'll be doing a lab on campus together in lab that will simulate this. So what is happening here? Well, let me explain the figure. Now, if we talk about an area, let's say your upper back, which is not very sensitive, and you'll see that when you do the lab with me on campus. Well, if we're in the upper back, notice that there's lots of different fields which have a sensory axon coming away from it. So for example, this particular axon here is sensing this field, and the green one here is picking up this field. So green with green and green over here with green. Now in an area like your back that is not very sensitive, look what happens when these neurons come together. A whole bunch of them work together with a single signal that goes to your brain, which means, hey, something in this area was stimulated, but not quite sure. Does it really matter on our upper back, or does it matter just to know that something is touching? Well, it turns out on our upper back, nature has favored just general knowledge that, hey, my upper back is being touched. Now, if we go to the fingertips, we see something very different. Each field has its own neuron and each pathway going to the brain. So if you get stimulated right here, the only pathway that gets stimulated is the one from that field. And same over here. Now the field is not infinitely small. There is still a field, So anywhere in that field that you stimulate, you would sense it as the same point. So let's talk about how does the clarity of this get enhanced. So this is called lateral inhibition phenomena. What it does is increases the granularity or sharpness, no pun intended with the pin here, but the ability to discriminate different points. So for example, that's really important in Braille. You have to be able to determine the two bumps right next to each other. And that's only possible because of the high number of sensory cells. So let's talk about how does the brain increase sensitivity, increase granularity, sharpness in this. So for example, if you took a pen or a pencil, or anything pointed, and you pushed it onto your skin, there'd be more stimulus here, but there'd be a little bit on the edge as well as the skin is deflected. But notice that B is getting the most intense stimulus, A and C are getting less. And this is how it's would be coded for if you could sense it right there at the skin. But look at what happens in the secondary neurons. Because A and C are not sending as much neurotransmitter as B is, what B does is says, well, why don't you ignore these two on the side and focus just on me? So you can see these two things here actually tell the adjacent neurons, if they're not being stimulated very much, hey, don't send a signal because you're making it blurry. You're making the signal blurry. So what happens in the tertiary or third level neurons, only B fires. So even though A and C were actually stimulated, they get inhibited so that the signal looks like this at the skin, but feels like this in our body. Notice the lateral means on the side, lateral inhibition. These two, A and C, were told not to send a signal. B was told enhanced its signal. And so what we feel is different than what it is. We feel that the pin is touching us just here and that none of the rest of our skin is being deflected. So what that does is increasing our sensitivity and sharpness of sensation. It's called lateral inhibition phenomenon. There you go. All right, more about touch. So if there's free nerve endings, we would find that for both pain and chemicals. So pain is very famous for having free nerve endings, no specialized structure. Now, what is pain? Well, it turns out pain is damage to your tissues. You're sensing damage to the tissues. So either the neuron itself is being damaged or chemicals from the tissues that are being damaged are stimulating the pain receptors, sending signals down. The same with chemicals. Chemicals directly stimulate those dendrites, which sends a signal down the line. So Pacinian corpuscles use if you remember, I told you they use a enforced or encapsulated nerve endings. And what they sense are deep or steady pressure and vibration. So in this case, you feel pressure when you're holding on to something, but you can't use those to feel texture. Now let's talk about texture really quickly. If you put your hand down on some sandpaper, pretty much any sandpaper, right off the bat, you'll feel the little bumps. Fairly quickly, though, within a few seconds, you stop feeling them. So how do we get a constant information about how bumpy the sandpaper is? We move our fingers across it. We slide our hands across surfaces to get texture, because as those little bumps are changing places with each other across our skin, the skin never has a chance to adapt. So if we want to check texture, we don't just put our hands on something. You're not going to get much from that. You can try it. Something that has texture. Put your hand on it and hold it there for a moment, and you'll stop sensing the texture. But if you want to sense it again, you slide your fingers across and you're like, oh, it is bumpy, because you're not giving any of those sensory receptors a chance to adapt. The Pacinian corpuscles, on the other hand, are slower to adapt. And they're saying, are you still holding onto this object? How much pressure is on the hand? Are we going to drop it? Or are we going to let it go? Okay, Ruffini corpuscles are found in the skin, and they sense skin stretch. How much is the skin stretched out? And Meissner's corpuscles are those that I told you about for sensing texture, fine discriminative touch, light or brief touch. So it's brief because as you move your hand across it, those little sensory cells only get stimulated for a brief moment. They send their signals. They never really get a chance to adapt. And so we use these for most senses of texture. Temperature. Okay, I told you I would talk about the cannibal Watt phenomenon and the fact that we only sense changes. So it's a free nerve ending, just like we saw in pain chemicals. But what do cold receptors? We have cold receptors and we have warm receptors. We don't have any receptors for boiling hot or freezing cold, and I'll tell you why. So cold receptors only tell you that the skin is dropping in temperature, that the environment that you're in is cooler than the current skin temperature. So for example, let's say my hands are 75 degrees. If I put my hands in water that are 60, it'll feel cold, but it'll stop feeling cold as soon as my skin drops to 60 degrees. It stops feeling cold because my skin is no longer changing temperature. It's the same temperature as the water, so you don't sense any temperature at all. It just feels like water. Warm receptors, the same thing, higher than current skin temperature. And by the way, T here is temperature. So the question is, you could test this. So draw yourself a bath, whatever is comfortable for you, not too hot. And then get in. What do you first sense? You feel, it's warm, or maybe it's like a little hot, feels good. But if you sit in there for a while, it stops feeling warm. So how do you get that sensation back? Well, you have to add more hot water. What is going on? Well, it turns out that the bath isn't getting cooler. It's not losing its coolness. You could measure it. It's still within a degree or two of when you first got in, although the human body does cool the water off a little, not enough to compensate for what you feel. But what happens is your skin in the bath gets the same temperature as the bath water, so you stop feeling warm. Because remember, warm only happens when the environment is warmer than the skin and the temperature of the skin is rising. So if you want to have that sensation again, you add hot water. Now the temperature is warmer than the current skin temperature, and your skin starts warming up. If you keep doing that over and over again, you're going to pass out because you'll end up with too high of body temperature. And that's what happens in jacuzzis, hot tubs. And that's one of the reasons why we have laws in various states like California that says you can't go higher than 103 or 101. I can't remember. I think it's 101. Anyway, what they want to make sure it doesn't happen is, let's say you set your jacuzzi at 105 or 106. Well, I feel really good getting in, but what would happen is you would stop feeling warm. You'd stop feeling the heat. And then what would happen is your internal body temperature, because you don't really have any way to sweat, would go up to 105. And if you had a little liquor, you might just pass out, go under the surface of the water and drown in a hot tub. And that has happened. And that's why they don't allow 105-degree hot tubs in public jacuzzis, like at public pools and motels, hotels, etc. What is the cannibal pot phenomenon? Well, the idea is if you put people in a pot and heated it slowly enough, you would never notice that it was boiling, because the temperature increase would only be very, very gradual. So you could actually boil someone to death and they would never feel much pain. But what is that feeling of burning hot or freezing cold? Well, it turns out that when you add warm receptor to pain, so for example, if you got in a bath that was 120, that would hurt, because the warm receptors are sending signals as well as the pain receptors. Freezing cold, same thing. Cold receptors are being stimulated, as are pain receptors. And that's where freezing cold and boiling hot comes from. So pain receptors, excessive heat or cold, cause tissue damage and pain. There's no receptor for hot. If it feels hot, it's warm plus pain, and the same with cold. Okay, coding for intensity and duration. I made an allegory to this, or an allusion to this earlier. But we're going to talk about this figure. It may come up as a multiple choice question on the test, so please pay attention. So let's talk about the top, stimulus A. Notice that it's not quite as tall as stimulus B, and it takes less time. Duration means how long it takes, and that's the width. So if we take this actual stimulus and let it be coded by the nervous system, it looks like this, right at this. And then if you move a little bit further down away from the transduction site to the actual trigger zone where there's axons, remember there's going to be individual action potentials here. Each one of these spikes is one of those curved structures that you just finished talking about on the last test. So notice that the signal intensity was lower, so each of these action potentials is going to have a little bit of gap between them. And then if you get all the way down to the end, we're past the trigger zone, past, by the way, this is all, this rise and lower is a graded potential. The spikes are action potentials. So notice that you're not going to get a really big change in the intensity or the the frequency of axe potentials because it's not lasting very long. By the time you're about to adapt, it stops. Notice that there's very little acetylcholine released or other neurotransmitters. More interestingly, let's go to the longer and stronger stimulus. So notice that it's higher and it starts to diminish a little bit, but the key here is I want you to look at these two things. Notice two things. Notice here at the beginning, the axe potentials are really close together, meaning it was a much stronger, more intense stimulus. But what's even more interesting is look what happens towards the end here. They start spreading out. This is adaptation. This is what happens with olfactory fatigue. It starts to become less and less and less. And for some senses, it would go to basically to zero. But notice because it's taking longer, the gap is starting to expand as adaptation happens. So what's last thing I want to reinforce with you guys is that the axe potentials in all four places are exactly the same height, negative 100 millivolts. Now notice that they're going from negative 70 up to 20. It's 100 total. The height of all of these, regardless of how intense it is or how long it takes, is going to be the same height. The difference is how many are coming per second. Many more in the strong stimulus, fewer in the weaker stimulus. All right, pain and itching. So they both stimulate structures called nociceptors, and these are free nerve endings that pick up tissue damage that we interpret as pain. What is the reflexive pathway? Well, the sequence of structures that these signals go through to allow you to have a reflex to pull yourself away from the pain is we start with a sensory neuron that transduces it into the action potentials. Then the next spot is the central nervous system, usually the spinal cord, unless it is a pain reflex that happens in the head. So everything below your head, reflex is going to happen in the spinal cord, and then it goes directly to a motor neuron. Notice that no real decision has been made on the part of the brain, and then eventually a muscle. This could also be a gland. By the gland could be another end point for the pathway for reflex. What's the key? What it allows is, because it's a reflex, it allows a really rapid response without conscious control or inhibition. And that is, you're going to have the same exact response to it, regardless of whether you're paying attention or not, or whether you want to do it or not. And a good example of that, maybe not with pain, but you can't really, most people can't hold their eyes open when they put in contacts. They have to actually inhibit it with their hands. They have to hold their eyes open, stick the contact in, because the blink would happen as a result of the reflex. Even though your intention is to keep your eyes open, the reflex says, nope, this is a reflex and you're not going to be able to inhibit it. Why is adaptation not seen in nociceptors or pain receptors? Well, there's a couple of reasons why adaptation doesn't happen. Now, if you remember, adaptation is the diminishment of signal getting to the brain from the receptors saying, yeah, things are pretty much the same as they were, so I don't need to keep sending you the signal. So why is pain different? Well, pain is saying to you, hey, there's still a problem. You're still in pain. That means there's still tissue damage. So why doesn't it adapt? Well, if you adapted, you would stop thinking it was important and you wouldn't get away from the danger that is causing damage to your body. So the idea is, why doesn't it adapt? Well, it means that, hey, there's still a problem. But what if you get away from the problem? Why doesn't it stop hurting if you have an injury? Well, the pain of the injury away from the stimulus that's causing the injury is to say, hey, that body part is still injured. Don't use it. That's what that pain is for. But what is the last function of pain? Well, pain is there primarily afterwards. What is the function of having felt it for so long? Is to tell you, hey, dummy, don't let that happen again because you got hurt. So what pain is, because it takes so long, it doesn't adapt. It's a way to teach a really important lesson to your subconscious, to your brain, that says, don't let this happen again. Now, what are some exceptions to those pain lessons? Well, it's pain that is either really important and necessary, such as childbirth, or pain that we choose. Now, what are some pains that we choose to do? Well, think of tattoos. Tattoos are something that your brain doesn't say, whoa, 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 don't let that happen again, because you chose to do it. And you know that the damage is minimal. Another pain that you choose is hot sauce. It's really hot, it's going to hurt. But it doesn't make you not want to do it again. It just makes you think this is part of what happens with this. And the same with childbirth. It hurts really, really bad because we get a baby out of it, and because it's so important biologically, our brain doesn't say, don't let that happen again. Babies wouldn't be born. All right. The scientific term for itch is pruritus, but please notice it's also going through those same receptors as pain. So when you itch, the signal is going through the same pathway as pain. It's caused by nociception. So the question is, what is our response to pain, to itch? It's to scratch it. So what is the benefit of scratching an itch? Well, what it does is it acts as a sort of defibrillator. You guys know defibrillators overwhelm the heart, get it back onto its normal rhythm. Some scientists believe that scratching an itch is kind of like a defibrillator overwhelming the nociceptor with signals and resetting it back to normal. Now, you can test this. If you feel an itch, you scratch and it feels better because now you don't have the itch. But if you scratch just as hard on that same body part, when it doesn't itch, it hurts. That's because you don't need to defibrillate that itch. The only time that doesn't work is when there's a chemical in your skin that's kind of causing continuous itch. For example, when a bug bites you, like a mosquito, that chemical's still in there, and so scratching doesn't help because that defibrillation is not going to end the stimulus, which is the chemical that's under your skin. Mechanism is unclear. Most treatments are ineffective. Some people get some benefit out of hydrocortisone, but I personally really like Gold Bond. Gold Bond does a great job with itch. So if you do have chronic itch, I used to have a lot of chronic itch, and I only found Gold Bond effective. Most of the cortisone creams didn't work for me. Why is chronic itch so disturbing? Well, it turns out that it's one of the few sensations that you just absolutely can't ignore. And the classic is you've got your arms full of groceries or something that you can't drop, and your SOB nose decides right at that moment to put an itch in your nose. And it's really hard to just say, well, I'm just going to ignore it until I get inside my house. It becomes so overwhelmingly motivational. And I believe that that's because itch often is a sensation of a bug crawling on you or something that really needs to be addressed. And it's really not exactly clear why that's the case, but itch is one of those things that you really can't ignore. And even though pain doesn't adapt, many people find ways to ignore pain. They can put it in a certain part of their brain, but that's not the case for itch. Itch is one of those things that you really feel obligated and you have to scratch it. It is an obsession, a compulsion to scratch. All right, referred pain. Okay, now you guys got to listen to me because you're going to get this wrong on the test if you don't listen. When I say what is the reason for referred pain, it is not ischemia. The cause of the pain portion of this is ischemia. But the cause of referred pain is something different. I'll be talking about what is the cause of the referred pain and why is it different than other kinds of pain? So the answer to the review question, what causes referred pain, is not ischemia. Make a note of that. Make a big note in your notes. And so when you come back to this for your review questions, the answer is not ischemia. That is the cause of the pain portion of it. But when I'm asking for the cause of referred pain, it has to do with nervous pathways, not ischemia. So make a note. All right. What causes it? Well, you guys have probably heard about the fact that when you're having a heart attack, sometimes you feel pain in your left arm. Is that where your heart is? No. But it turns out that the pathway for your left arm up to your brain and the pathway from your heart to your brain use the same sensory nerves. Why would the body do this? Well, it turns out that because your heart almost never has a problem, that the brain or nature decided not to devote a pathway of its own from the heart to the brain because, if you're not going to be using it very much, why dedicate a completely dedicated line to just that structure? It's generally never going to have a problem. But what it turns out is that we have lots of signals coming from our arm. What happens is because there's a convergence, that means two become one, that once in a long while, the heart takes over and sends the signal up that pathway. And if you don't know what it means, pain in my left arm for no good reason, you could die because you wouldn't get help from the heart attack. And so it's just a way of kind of killing two birds with one stone sort of thing. And we're going to see the same thing, believe it or not, in our throats, between our trachea and our esophagus. Why would this cause referred pain? Well, the reason why it's referred pain is because our brain is so used to getting signals from the skin of our arm that when it comes from the heart, it just assumes that it's still coming from the arm because it's coming up that pathway. Remember, there's nothing in the signals themselves that says where it's coming from. So all the brain knows is, hey, this pathway that I'm used to feeling from my left arm is being stimulated. So that means I have pain in my left arm. But we as modern humans know that when you feel that without a good reason, hey, that's a clue that you might be having a heart attack. We understand that. So we say, we reinterpret that as a potential problem. Okay, what are pain-reducing drugs? They're called analgesic drugs. So when you hear that term, an analgesic, it means any drug that reduces pain. The most common and cheapest are aspirin and ibuprofen. One of the main reasons they work is they reduce swelling and slow transmission of pains from the injury site. Question, why is aspirin so darn cheap? Well, it's one of the reasons. The reason for this is why drugs are expensive. It's the idea that you can create a chemical, you can patent it, and then other people can't sell that exact molecule because you created that molecule in your laboratory. Why can't you do that for aspirin? Well, it turns out that aspirin comes from a natural source. What natural source? I'll give you a hint. Native Americans, hundreds of years ago, used to strip willow bark off of willow trees and keep it with them. If they found themselves aching or hurting in whatever way, they would chew on the willow bark and it would get better. So what was the chemical that was in that willow bark? It's called acetyl salicylic acid. We call it aspirin. And because it grows naturally in willow trees, you can't patent it. And so that's why aspirin is so inexpensive. It works well. It comes from a willow tree, can't be patented. Ibuprofen can. It's still fairly inexpensive because the patent for it expired and there's generics, et cetera, et cetera. All right, opiates are very different though. They block the sensation of the pain in the brain by blocking neurotransmitters. So for example, morphine is an opiate, Vicodin, a variety, you guys know the ones I'm talking about, the ones that are really powerful analgesics, pain blockers. Now, no one's going to get addicted to aspirin or ibuprofen, but they can get addicted to opiates because what happens is, if you remember, I talked about a while back, we talked about downregulation, the idea that you stop sensing it as much. Same thing happens. It takes more of that same opiate to have the same sensation because the sensitivity diminishes over time. And that's why when people overdoses, what they're trying to do is get that same original sensation of either pain blockage or euphoria, and they have to take more and more and more. And eventually the toxicity of the drug kills the person. And that's why opiates are very, very dangerous and should not be used for long periods of time, except as very closely monitored by a physician. So you guys know the opiates we're talking about. Any of the opioid things like codeine. I talked about some of those. All right. So here's an image from your book showing you how referred pain works. So because there's a similar pathway, look over here on the right, the pathway between your kidney and your skin share the exact same path. So when it goes up to your brain just assumes it's coming from your skin because kidneys almost never have a problem. But when they do, it feels like the sensation is coming from your skin. So basically lower back or abdominal areas where you feel it. But the most interesting for me is your spleen coming up onto your shoulder and your heart. Now your heart is found right about here. So part of it makes sense, but going all the way down your arm doesn't make sense. So instead of this being your kidney, imagine this is your heart. This is the skin of your left arm. Comes together, it synapses with a single secondary sensory neuron. So all your brain knows is the pathway that normally sends signals from your arm, which is a constant. It's sending a signal, so shoot, that must mean my arm is in pain. It doesn't. So notice that many of these kind of make sense. The appendix is kind of right around there. Urinary bladder is right around there. Liver and gallbladder, the thymus, which is down here around your chest, refers up here. Spleen, that's a weird one. Your spleen is way down here. and it refers right up to your shoulder. So if you feel pain in your your left shoulder, which is our right, there it is, left shoulder, right on the tip, it could be a problem with your spleen. All right, sense of smell. Olfactory cells convert smells into action potentials. What is the functional category? Chemoreceptor. Olfactory cilia branch out into mucus and receive odorant molecules. The olfactory bulb and tract are found at the bottom of our brains. The ganglion below the frontal lobe are thicker because the cell bodies are there. And the axon sends signals towards the brain. So I'm going to show you all how these olfactory cells work. Olfactory is linked to the limbic system. What is the limbic system? Well, the limbic system is involved in emotion and long-term memory. So you may have noticed that if you smell something, it'll bring back memories from distant past with real intensity and vividness, clarity. That's because smell is linked to long-term memory and emotion, which also adds to memory. Why do we feel such strong emotions when we smell certain odors? Because the limbic system, which is tied to olfaction, is responsible for long-term memory and emotion. And emotions reinforce memory. So certain smells can take you all the way back to when you were a child and bring up all of the things that you were feeling and all the things you were seeing. It's a much more evocative sense than pretty much any of the other. Okay, so how does this work? How does smell work? Well, we're smelling coffee. It goes up into our olfactory epithelium, which is way up here. See, right, this little square? This is where it is. These nasoconcha are not about smell. It's right up in this little tiny square here, right up in this area here. So if we were to look in this little square, we would see the odorant molecules coming up and sticking to the mucus. They diffuse through the mucus and they hit these little olfactory hairs. The olfactory hairs stimulated, and that sends a signal all the way up through the little holes in the ethmoid bone. This part of the ethmoid bone is called the cribriform plate. If you've ever had anatomy, you know there's an area on either side of the crista galli. It's very thin and full of little holes. There it is, the ethmoid, the cribriform plate of the ethmoid bone. And then this area is swollen because we have mitral cell bodies, which make it thicker, and it sends it then through the olfactory tract, which is a nerve running back from the olfactory epithelium to the brain. But here's what's interesting. Notice that these olfactory receptors are right embedded in the actual air, only protecting a little bit of mucus. Most sensory receptors are isolated from anything that could cause damage. Now, because these olfactory receptors are right there on the surface, any toxic chemical, for example, chlorine gas, could kill them. And so they have to be replaced very often. So these cells are very quickly replaced. And then these other cells on the side are helper cells that reinforce them and help them divide and make the pathway down to the olfactory cilia. Okay, taste. This is another image from your book. There are 5 tastes. We used to think there was only 4. Sour, sweet, salty, and bitter, but it turns out that there's a last called umami. And I say it a little different just for fun, umami. And that's deliciousness. And we get that in one way from MSG. And that's one of the reasons why they put MSG in things like cup of noodles, is so that you will think it tastes better than it actually does because it's so darn cheap. We think it tastes great, not because they put delicious stuff in it, but because it contains MSG, it tricks our brain into thinking it's more delicious than it is. Now, please note, taste buds are not on the surface. These are called papilla, lingual papilla. The bumps are not the taste buds. But if you go down into these creases between these buds, between the papillae, you see that these individual taste buds are found on the side, not on the top. But why is this significant? Because much of the food that we taste is very rough, hot, and by having the taste buds be down in these little grooves, it isolates them somewhat from damage. Now, if you've ever been wine tasting or wanting to taste something that's different from something else, when you taste wine, you're supposed to swirl your mouth out with water. Why is that? Because some of the wine is still embedded down in here when you take your next drink of wine. And the old wine is going to mix with a little bit of wine that's still there. When you swish with water, it washes the wine that's in this groove out. So when the new wine comes in, you're tasting only the new wine and not the old wine. And so if we look at an individual taste bud, we see that there's taste hair sticking out, and basal cells, gustatory cells. By the way, gustar is Spanish for to taste. So gustatory and gustar are related. Transitional cells are the cells that are part of this as well. Okay, Now we're going to be talking about how hearing works, and then I'm going to transition real quickly right into how vision works in the next chapter section. The ear is a sense organ that's specialized for two functions, hearing and equilibrium. Now we're going to focus just on hearing for now, and these are the main structures involved. So if I ask you, and I'm probably going to ask you, how does hearing work? You need to tell me about all these structures, starting from the auricle or pinna, going through the ear canal. By the way, all of this here is external ear. Everything here in red is middle ear, and everything in gray and yellow is inner ear. So sound waves come, they are gathered by this portion of the auricle. We're going to be talking, what is this part on the edge for? Why do we have it? Why is it shaped so differently from a cat or dog ear? Anyway, sound waves get funneled in here. They strike the tympanic membrane. The bones move. That causes fluid in here to move. When hair cells in here get stimulated, signals go out through the vestibular nerve for the vestibule, but the cochlear nerve for hearing. So we're not going to focus on the vestibular nerve for now. We're going to focus on the cochlear nerve, because the cochlear nerve is what's going to send sound. Vestibular nerve sends signal about balance. So we're going to ignore the vestibule, semicircular canals, and vestibular nerve for now, because we're talking just about hearing. Okay, so these are the anatomical structures. Let's get on to how it works. Sound waves act as an indirect stimulus. So the sound waves themselves are not going to stimulate the hair cells that are going to create a second, or really third stimulus that will eventually generate action potentials and second order hearing cells. So the sound waves come in, they're basically pressure waves, area where there's more compressed air, and areas where there isn't. That's what sound waves are. So the conduction mechanism is in the air through the outer ears, your pinna, external auditory canal. It strikes the tympanum. So what does the tympanum do? Is it converts pressure waves into mechanical vibrations. So imagine that you were standing on a dock in a lake and you see waves coming towards you. Well, what is going to happen as a result of the waves coming? If they're medium or small, the dock starts rising up and down. So what you've done is you've converted the waves on the lake to movement up and down of the dock. Well, the tympanum does this too. So pressure waves are converted into physical movement forward and backward, the same way the dock moves as a result of the waves on the lake. Now, the tympanum is connected to these middle ear bones. They're not inner ear bones, they're middle ear bones. And they're called the malleus, incus, and stapes. Sometimes we call them hammer, anvil, and stirrup. In my class, you need to use the scientific terms. So what happens? Now that the tympanum is moving, the middle ear bones conduct the vibrations to the inner ear. So why do we have these? The middle ear bones have an important function. What they do is they help amplify the strength of the movement while sacrificing the amplitude. So they increase the strength but decrease the amplitude. In addition, why do we have middle ear bones? There are little muscles attached to them that allows us to block the vibrations coming in so that really loud sounds don't cause us too much damage, as long as that the intensity is fairly continuous. So we can actually stop hearing loud sounds if they last for more than half a second. The next stage, the middle ear bones press on what is called the oval window, and these oval window then generates fluid pressure waves. So we went from air waves to mechanical vibrations, now to fluid pressure waves, kind of like waves in the ocean. So we're all the way back to waves again. And because the oval window is connected by these bones, you have to press harder on the fluid than you would in air to get the same movement. So that's another reason for the bones, is they are able to make these movements happen in liquid, which is much more dense than air. So the next thing is these fluid pressure waves go past these membranes. One is called the tectoral membrane. And I'll show you how this works. It basically presses against little hairs sticking out of the hair cells, causing them to flex, and that sends what that generates, what's called a receptor potential. I'll talk about what happens after that. The tectoral membranes contact the basilar membrane. One of the things that's not mentioned here is the basilar membrane. So we have a tectoral membrane and a basilar membrane. They work with each other, one pressing against the other. So the hair cells have these little hairs sticking up called stereocilia. when the tectorial membrane presses down on the hair cells, which are part of the basilar membrane or organ of Corti. The flexion of these hairs cause depolarization of the cells. That generates a receptor potential, kind of like where we saw a graded potential. So a receptor potential is kind of like a graded potential, but the difference is we're not building a receptor potential up for an action potential. Basically, as the intensity, more sodium comes in, more of the hairs are bent, more acetylcholine is directly released. So there's no axon hillock, there's no threshold over which you have to cross in order to start sending neurotransmitter. So there's not that, hey, only send a signal if it's strong enough. It will actually send a little bit of neurotransmitter if a little bit of the hair cells are bent, because a little bit of sodium came in. Okay, so how do we perceive where sound comes from? Well, there's two ways. First of all, I'm going to talk about what sound is. Remember I told you sound is basically pressure waves. So if we were to see sound, we would see that the air molecules are more compressed in some areas and less compressed in others. So these are like waves in the same way that the height of the water is higher here than here. This would be a crest and a trough. This is more compressed air, less compressed air. So back to that riddle. If a tree falls in the forest and there's no one there to hear it, does it still create a sound? Well, it depends on what your definition of sound is. If does it mean that does it still create the things that we would be able to sense as sound? The answer is yes. But sound really is the interpretation of these pressure waves by an organism with an ability to hear. So does it make a sound? What is a sound? Well, it turns out a sound is not the same as these pressure waves. So the pressure waves and sound are not the same. Sound is really our interpretation of these pressure waves through this complex mechanism of our middle and inner ear. So the real answer is, if a tree falls in the forest and there's no one there to hear it, the answer is it does not make a sound. It makes pressure waves, but it takes an organism like a person, or a mammal, or anything that can hear to convert those pressure waves from the tree into something, into sound. So from a physiologist's point of view, if there is no one there to hear it, animal, human, whatever, it does not make a sound. It just makes these pressure waves. So there you go. All right. What is the difference between these two figures? Well, it has to do with the frequency of pressure waves. So we can see the intensity is the height of the waves is pretty much the same, but in this case, the frequency is lower. Amplitude in decibels is how tall the waves are. And here the frequency is higher. That means there's more waves coming in a shorter amount of time. We saw that that was a way to code for intensity. Now, it turns out your body does not use the frequency of these things to code for the intensity of them. So it gets weird, so you guys got to pay attention. Okay, so here is the figure from your book. And this is the figure, or one very similar to it, is I'm going to be asking you, how does our ability to hear sound work? So let's start at the beginning. This is an essay, and it's worth 10 points, by the way, guys. So it's really important that you pay attention and take really good notes. Okay, so these are the sound waves. This is not sound. These are pressure waves. And you can see that they're this far apart. And they're making their way in and striking the tympanic membrane. So they enter the ear, the external auditory canal, and they strike the tympana. If you remember, in response to this, in the same way that dock waves on the ocean or lake hit it, it starts moving back and forth. So the tympanic membrane vibrates in response to the sound waves, or pressure waves. Better to call them pressure waves, because they're not really sound yet. It doesn't become sound until, believe it or not, it gets all the way to your brain. In the next step, these little bones start moving. And remember, we have these bones to increase, it says vibrations are amplified. So even though the overall distance that they move is diminished, the strength of the movements increases, so amplifies the movements. The other reason we have them is there's a place right here where I'm pointing to, and right on the stapes at both ends, where you can attach muscles and pull those muscles to keep the vibration from happening, which then prevents the cochlea from being overwhelmed. Now, step 4, these vibrations at the end of this, there's that little oval window, and there's the stapes or stirrup. We're right here. It starts pressing inward in the same frequency as the vibrations were coming in from the air. But instead of air waves, now it's fluid waves. Fluid waves start moving their way in. And if you know anything about how fluid is, you know that you can't make it any smaller. You can't compress fluid the way that you can compress air. So if we push inward, there has to be a place for it to go outward. So inward is the oval window. And since this fluid has to have somewhere to go, it presses outward at the round window. So every time it pushes in, this part bulges out. So what's happening is kind of sloshing back and forth. And the sloshing back and forth are these fluid waves that then start stimulating the basilar membrane because it's hitting the tectoral membrane, flexing those little hair cells, which I'll show you in a moment. That sends the signal out saying, hey, there's sound, because these little hairs are bending as a result of these waves coming through. So pressure bends the membrane of the cochlear duct at a point of maximum vibration for a given frequency. Now, here's what you need to know. It turns out that whether the pitch is high or low has nothing to do with how many of these signals are coming out. It has to do with where in the cochlea is being stimulated. So you're going to need to include that in your essay on how does sound work. Okay, so here's another figure from your book. So we saw those steps. How does it get in here? And how does it put pressure on it? Well, how do we determine whether it was a deep pitch sound way out here or a high pitch sound? Well, the way that this works is very much like a piano keyboard. If you've ever played piano, you know that at one end are the high pitch sounds. I want to say far right is the high pitch notes, and the far left are the low pitch notes. It has to do with how long and thick the chords, the strings are inside the piano. Well, in this case, if it's a very low pitch sound, the only part of the cochlea that will move if we unwound the snail shell out to be a straight line would be near the apex or tip. Remember, the sound waves are coming in, or the pressure waves are coming in this way and out that way. But if this part, the apex, the end, starts to move and send signals, your brain interprets that as low pitch sound. So for example, thunder or a deep voice would stimulate this part of the cochlea. If it was medium pitch sounds like the human voice, this part of the cochlea would be stimulated. Notice that this is the frequency of vibrations that would stimulate it. If it's very high pitched sounds, it's going to stimulate the area right near the oval window. And notice that the length of the fibers, the longer the fibers, the less they're going to move. So here, they're very near, the fibers are very short. Here at the end, the fibers are very long. So this will only move with low pitch sounds, medium pitch sounds, and high pitch sounds. In the same way, you can't hit one end of a keyboard and make it sound like the other end of the keyboard, no matter how you hit it. It's all the location. So your brain knows it was a high pitched sound because this part of the cochlea was being stimulated and sending signals to that part of the brain. The brain knows it was a low pitched sound because this part of the cochlea out near the tip was sending a signal. But the question is, how loud is it? Well, here's another figure showing you basically the same thing. I don't know which of these two figures is going to be on the test. That would be this one or this one, but they're showing basically the exact same thing. Remember, high-pitched sounds here, medium-pitched sounds in the middle, and low-pitched sounds at the end. Saying the exact same thing, but with a slightly different. But here I kind of like because it actually shows those pressure waves then being converted to mechanical waves, and here it's being converted to fluid waves. Okay, so how does it work? And how does intensity or volume? Well, fluid waves move along the cochlea when the oval window is pressed and vibrated by the staphes. This was step 4 in our first figure. The tectoral membrane vibrates as a result. This is the membrane that's on top of the basilar membrane. So the stereocilia that are sticking out of the basilar membrane flex when the tectoral membrane presses against it. And when they flex, something happens. That causes ion channels to open, usually sodium channels. And that causes the membrane potential to change. And you get depolarization. But remember, it's not the same depolarization that you would get in an axon. It's a different kind of depolarization. This is more like a receptor potential rather than an action. But the key is, as you depolarize more, MT as neurotransmitter is released between the hair cell and the sensory neuron. Notice the hair cell is not a neuron, it's a sensory receptor. So it's going to release acetylcholine or other neurotransmitter, and the sensory neuron then is going to start going through graded potential up to an ax potential, and then send the signal. So here's the next question. How do you know how loud the sound is? The more neurotransmitter that gets released, the more ax potentials are going to be generated, and there's going to be more ax potentials per second. If you remember earlier in the lecture, I said more frequent ax potentials tells the brain that that was a really intense stimulus. So we code for volume by how many ax potentials there are, and we code for high pitch or low pitch on where on the cochlea the signal is coming from, what part of it is getting stimulated. So for the frequency of sound, it's location. And when it's the intensity of the sound or volume, it's the frequency of ax potentials. This can be very confusing. So here's the last step. Ax potential sends signal to the brain. And in the brain, those ax potentials, where did they come from and how many were coming? Tells us what pitch the sound was and how loud it was. Okay, so we're going to go into a little bit of the detail now of, remember I said I talked about the basilar membrane, the tectorial membrane, and these fluid-filled canals. When we take a cross-section through the cochlea, we see that there are 3 tubes. 2 of them are named in Italian, scala tympani and scala vestibuli, and then the cochlear duct is in the middle. It's sometimes called the vestibular duct and the tympanic duct if you don't want to do Italian. Well, what's really happening here is fluid is sloshing through here and coming out here. But the key is when that sloshing happens, this whole system starts to move. So if we were to look right at this system, there's the tectorial membrane on top and the basilar membrane on the bottom. Notice that the tectorial membrane is crashing down on top of these stereocilia. When that happens, when they bend, ion channels open, and acetylcholine right in here gets released. And when the acetylcholine gets released, then ax potentials in the second order, or really first order sensory neurons, get sent out. This is where the first ax potentials happen. So when ion channels happen here, open here, we get this what is called a receptor potential increasing. And as receptor potential increases, so does the amount of acetylcholine or other neurotransmitter between the hair cell and the first order sensory neurons. Okay, so I talked about this. So how do we determine the pitch of the sound, whether it's a high pitch sound or a low pitch sound? It's where on the basilar membrane it is being stimulated. So location is everything for pitch. How do we determine intensity? How loud the sound was? It's how many ax potential are coming per second. In the same way we were able to determine how hard we were being pressed on the skin, we can determine how intense the sound is, volume, by how many ax potentials are coming. I hope you guys can get this. It's tough. Remember I told you localization determining directionality of sound? I'm going to be talking about that on the next slide. What are the different types of deafness? You can have conductive deafness. That is all the mechanical stuff. The tympanum doesn't move when pressure waves hit it. The ossicles won't vibrate. In otosclerosis, as we age, the ossicle joints become more rigid. Sound doesn't get transmitted as well. We can't hear as well. Or anything in the cochlea, because that's still mechanical. That's still something moving. In sensorineural or central, sometimes called nerve deafness, is the inability to convert the vibrations into ax potentials. That is something in the hair cells, or the inability of the signal to get all the way up to the brain, or even interpret the signal from the cochlea as sound. So anything having to do with nerve cells, the sensorineural or central, sometimes called nerve deafness. And conductive deafness has to do with the mechanical component. Remember, it goes from mechanical to sensorineural right at the hair cells. Okay, so how do we determine sound and why are our ears set up this way? So take notes again. So how do we know whether a sound has come from our left or right? That's a really easy one, believe it or not. Well, if the sound, like a trumpet, is coming from the left side, there's 2 ways that we know that it was on the left versus on the right. The first way that we know is it's louder on the left side than on the right, because the sound is basically closer and it's not being blocked by your head. So it's louder on the left if it's coming from the left. In addition, sound enters your ear slightly before on the left side than it does on the right. So because it comes first, and then the right, your brain says, aha, it must be coming from the left. So there's 2 ways of knowing whether it was left or right. how loud it is. It's going to be louder on the same side, and it's going to come in first on the same side. But the question is, how do we know up and down? How do we know if something's coming from in front of us up above or in front of us down below? And it has to do with the funky shape of our ears. Now, please note, this area that I'm highlighting right here is the only thing that funnels sound into our ears, the focus that functions very much like the ear of a cat or a dog. All this other stuff, all these little whirls and bumps give us directionality. Now, it doesn't really give us left and right because we already have that from this method. But what it does is it tells us whether the sound was coming from up above or down below us. How does that work? Well, if we don't tilt our head and we have to be looking at something intensely, and it's dark, and we can't see where the sound's coming from, how do we know if it's above us or below us? Well, here's the deal. It turns out that in the same way that sound goes in on the left side first and then the right, in an individual ear, the main part of the volume goes in through the hole and all the way down through the system. But in the same way that echoes in a canyon can happen after the main sound of your yell, little echoes from these little ridges and folds follow the main sound and give a code to the brain that says where the sound come from. So for example, if the sound came below you in front, I would come up and go into the main part, but then the shape of this is going to create a very unique echo pattern that's going to follow the sound in and tell your brain it came from below. We learn this by looking at the sound, remembering what does that echo pattern mean. But if the sound is coming from up above us in front, the sound's going to bounce off differently shaped twirl and curves. So the echo that follows the main sound is have a completely different code than it would have if it had been coming. So I do this with my students. I will do this with you too, especially if you remind me, please